Welcome to Study Loop.
For this question, we're looking at the process of phosphorylation and its effects on gene regulation. So phosphorylation is referring to the addition of a phosphorus group, typically onto a protein structure. So for example, phosphorylation can occur to our ribosomal RNA units. These are going to include a special subunit to them that we would call the eukaryotic initiation factor. And this is responsible for initiating the translation, which is the role of our ribosomal RNAs. How phosphorylation comes into play is a phosphorus molecule can be attached to this initiation factor, and this will stop or deactivate translation. So this is responsible for preventing the excess production of proteins or for stopping the production of certain genes we no longer want. Phosphorylation does not only have this deactivation effect, but it can also activate certain proteins. And these often play a role in signaling inside of a cell. So you may have heard of the G-coupled protein receptor, which is going to receive a signal from outside of a cell, and it's going to propagate that signal inside the cell using a signal cascade. And this relies on the phosphorylation of certain proteins inside of the cytoplasm. And it's basically activating them to play their role. So here, knowing that phosphorylation is typically for proteins and that it can either activate or it can deactivate these proteins and their effects, the best answer choice is going to be A. That these proteins can alter translation, they can affect the transmission of the RNA molecules outside of the nucleus, and they can have these other effects such as altering the ribosomal RNAs and transcription and translation of proteins.